The overall goal of the following experiment is to produce cross-sections of yeast communities to examine the spatial distribution of different fluorescent cell populations within a community. This is achieved by first freezing the community and then fixing it using methanol to preserve the spatial distribution of cells in the community. As the second step, the methanol is evaporated to allow penetration of the embedding OCT compound. The embedded community is then frozen in preparation for cryosectioning. Next, a cryotome sections the community and cross-sections can be imaged under a fluorescence microscope. Results are obtained that show the spatial distribution of fluorescent cell populations inside a community.
The main advantage of this technique over existing methods like optical sectioning is that it reveals the spatial distribution of populations deep inside yeast communities.
To begin this procedure, grow yeast communities on a membrane filter. This protocol corresponds to a typical community of less than 2 × 10⁸ cells on a 6 mm diameter membrane filter. Next, in a -20°C freezer to preserve a greater degree of fluorescence, use a piece of Whatman 541 filter paper to cover the bottom of a 1 cm diameter well. This filter paper is used as a carrier to transfer the community in later stages. Add 1 milliliter of -20°C 100% methanol to the well and leave the well at -20°C for the temperature to equilibrate. Now, freeze the yeast community by using tweezers to dip the membrane filter with the community in liquid nitrogen for at least 15 seconds. Next, transfer the frozen community to the well with -20°C methanol in the freezer. Ensure that the community is kept horizontal when submerging it in the methanol. Wait for 20 minutes.